The effect of temperature between gibberellins and auxins, analyzing growth using model organism Physalis vulgaris. For many decades, biologists have studied the relationship between environmental stressors, like change in temperature, with other species and organisms. Plants have physically adapted over the course of evolution to provide reproductive success. However, one of the two most important ways that plants respond are geotropism and phototropism, which require a constant balance between auxins and cytokinins that both determine growth in different ways for the plant. Besides growth, auxins also promote vascular differentiation to slow down how fast blades fall off the stem. And this is called leaf abscission. So here, this is in the apical meristem where the calyotyle receives the light. There's lateral distribution of the auxin to the shaded side. So the auxin move over due to the concentration, where there's a higher concentration, now more growth on that shaded side, resulting in a curve toward the right. Growth proceeds in a proportion to auxin concentration. So it'll grow in the opposite direction in which auxin travel to. There are lateral meristems as well as apical meristems, and those cause secondary growth. What was just explained was primary growth, which occurs at the tips of the shoots and the roots and produces new leaves and flowers. But lateral meristems cause secondary growth, meaning widening the plant, the circumference is going outward. This illustrates apical dominance, which is very important in the plant as the terminal bud is connected to the stem and it releases auxin, which inhibits the growth of the lateral bud. Removing the terminal bud it's called selective pruning, this process, but it allows dormant lateral buds to develop and it produces a way bottom heavier plant. The relationship between our internal physiological processes and environmental influences by Gloria K. Moody, and she said that in Arabidopsis roots, both of these polarities of IAA or auxin movement have been detected and linked to distinct physiological processes. Auxin transport is regulated and changes during development and in response to environmental stimuli. This brought us to our research question. This led us to try and compare the hormone auxin and gibberellins to analyze the change in plant growth. Excuse PPO, that was a typo. We are planning on injecting the specimen under the area of cell division in the area of elongation, where those apical meristems are producing those somewhat stem cells being ready to be differentiated into specialized functions. In order to test this question, we equally distributed sunlight to 5 plants and randomly selected which 2 would be incubated for 7 days at 80 degrees Fahrenheit or 26 degrees Celsius. To keep the only factors being tested, temperature and humidity, it was important to keep the same type of soil for the same species and age of plant, and the amount of light being exposed. Our second treatment group includes 3 plants and therefore 3 replicates in the incubator at a lower temperature of 26.7 degrees Celsius and a lower humidity of 65%. Figure 1 shows inferential statistics quantifying effects of plant growth in terms of hormones interacting with other hormones, such as the auxins and gibberellins interacting in terms of temperature and humidity in the model organism we're studying. So I know that a rate of a reaction, there's a denaturation of a protein that enzymes can lose their active sites, and it occurs at a temperature above 50 degrees Celsius, I believe, and that rate of reaction will go up on like a hill and it'll come back down. That reflects our findings here because